It should be started now, so go ahead.
Thank you. So welcome to the third webinar in the Bridge Sweden webinar series. My name is Malin Widerholm and I work at Stuns in Uppsala, where we focus on collaboration and innovation within life science. Before we begin, I would also like to briefly introduce the Bridge Sweden project that is behind this webinar series. So Bridge Sweden is a national initiative funded by SweLife through Vinnova, and the project runs from 2024 to 2026, and it aims to strengthen the development and production of biological products and pharmaceuticals in Sweden. The main goal of this project is to help other projects move faster from early discovery into the development, scale up, and eventually toward clinical application and patient treatment. So we bring together research infrastructures, universities, innovation offices, and industry partners to create a stronger platform for biologics and advanced therapies. As part of the project, we also are running 3 pilot initiatives that help strengthen the development pathway for biological products in Sweden. So this webinar series is designed for researchers, startup, scale-up companies, and organizations within the innovation support system who are working within biological products and pharmaceuticals. And through these webinars, we want to share practical knowledge, real experiences, and useful insights from across the life science ecosystem. And today we are very pleased to welcome Matilda Bäckberg and Olivia Merinder from Legado Bio. And they will talk a little bit about how in vivo studies support preclinical drug development and how these studies help guide important decisions before moving into clinical programs. And I'm really looking forward to today's webinar since preclinical in vivo studies are such an important part of early development. The presentation will be around 30 minutes, followed by a Q&A session. So please feel free to write your questions in the chat during the presentation or ask them directly afterwards. So with that, I would like to warmly welcome Matilda and Olivia. The floor is yours.
Oh, thank you, and everyone that has joined. So I will start to share my part of the presentation and see if it turns up for you. And thanks for the reminder. We're going to speak for 30 minutes. We will do our best to keep the time. So I hope you all see it now, the presentation, right?
It looks good.
So I'd like to start a little differently. Instead of that you usually do in the beginning with slides, you give a background of us, it will come, and the company. But I want to show you something that really represents how we think about our in vivo work. And what you will see now, it's an example of a refinement methodology developed by a former colleagues of mine. And to me, it illustrates the good in vivo work should look like. It should be good for the animals, good for the staff, and ultimately good for the sponsor and for the quality of the results. So I will now introducing you to the most important colleagues of us. So in this particular video, you will see mice coming when called. And as you see, they are very social and active animals. And some of them are keen to reach the sweets that are on the table to the left, and others just like the climbing. And these mice have temporarily left their cages and been released into what we call an observation box or play box. So when lab work are refined, when animals are handled well, and when the staff have the right conditions, they do an excellent work. You can reduce stress, improve the consistency, and generate better data. So that's the mindset that we want to have at Legado Bio. So just briefly introduce who we are. So I'm Matilda Bäckberg. I'm a researcher and the CEO of Legado Bio, and my background is in preclinical in vivo research, where I previously managed an in vivo facility for smaller laboratory animals, together with a highly skilled team of about 15 animal technicians in Södertälje. And we have also Olivia Merinder, which is the CISO of Legado Bio. She has a strong experience in both study design and study execution. And also within the team, we have Urban Höglund, and he brings the extensive scientific strategic experience of preclinical animal research, and many of you may have already been in contact with him. He used to be the former CEO of Adlego Biomedical, and Adlego and Legado, you hear the connection between the words, and it's an anagram of Adlego. I stay with that. This is the start of the introduction of the presentation. I will hand over to Olivia, who will talk more about the scientific side and how and why the in vivo studies are performed.
Thank you. Thank you, Matilda. So you probably all recognize this pipeline showing drug development going from a target hypothesis to submission and to marketing of your compound. And in vivo work is actually really important across multiple stages of this whole drug discovery process, from hit to lead optimization, all the way through preclinical development and even at the clinical development stages, as you can see from these little highlighted mice across the phases. And at each of these time points, the key goal is to answer translational questions regarding your compound. Does the compound reach the right place? Does it have the desired effect? Is it tolerable? What doses should we give? What schedule should we give to take it forward to clinical trials? And all of the answers to those questions will help guide candidate selection, the dosing strategy, and any progression decisions that will help reduce uncertainty before going to the next step. So in the next few slides, I'll walk through these main questions, what they are and how we answer them, and how the studies are designed to answer these questions. And bear in mind, this is a presentation, so it's going to be looking very linear, going from simple studies to more complex studies. But as you probably know, in reality, drug development isn't linear. It's up and down, going a little bit all over the place. So these studies that we do, they're generally overlapping. They are repeated. You have to start over sometimes, and they're all adapted depending on what your compound is, what your modality is, what your indication is, and what data comes up. So if you can switch slides, Matilda. So the first question we tend to ask regarding our new compound that has only been tested in vitro is, can it be dosed? Can it be given in vivo? So the first study that generally tends to be done is to give a single dose of our compound, whatever it may be, to see if it's biologically tolerable. And typically this is done as a single dose study with close clinical observations of the animals, generally rodents, mice, or rats, with the goal of identifying what the highest feasible dose could be and what the maximum tolerated dose is. And these results are generally formulation or dose dependent. Sometimes these studies in general are skipped. Sometimes they're combined with pharmacokinetic studies or efficacy studies. They're not always standalone tolerability studies. And they come up on again and again in the drug development process. They can be combined with dose range finding studies, which are performed later on. So these are important studies, and they happen throughout the drug development process, basically, to show that our compound and our formulation is safe. So the next question that gets asked is, once we know it's safe and tolerable, the next question is, is it released into the bloodstream? Does it get to where we want it to go? How much is released in the bloodstream? How long does it persist in the bloodstream? This is a very, very important question, because if your compound doesn't actually go into the blood, or not enough of it, or if you have to give a dose that is equivalent to say, 16 tablets of a paracetamol-sized tablet to reach the bloodstream, then it's not a feasible compound and it's not worth going forward. So pharmacokinetics, these studies are key. They're relatively non-complex in their design. It's usually a single dose of your compound given in the administration route you desire, intravenous, peroral, subcutaneous. Regular blood samples are taken at specific time points after sampling, after dosing, and you measure the levels of compound via various methods depending on the compound, but often it's LCMS. And then you get a curve that is shown in this picture here, looking at the half-life and the Cmax and the Tmax and the area under the curve, which is equivalent to distribution or bioavailability even. And this is showing basically how that your compound reaches the bloodstream at a key concentration to be able to work. And these PK studies can actually, single-dose PK studies can actually be used to predict how a compound will work if you're given multiple dosing. I think you need to click, Matilda, once more. There we go. So this is a picture of a in silico modeling where we have asked the model basically what happens to the compound if we give it twice a day, or if we give it once a day, or if basically a certain schedule of dosing. And you can see how long it takes for the compound to reach steady state and how many doses are required. So this is a in silico model based on an in vivo model to be able to drive our dosing schedule and dosing strategy forward. So doing animal models with computer-based design helps drive the development process forward. You can go to the next slide. So once we found that our drug is tolerable and it reaches the bloodstream at a sufficient concentration, we also want to answer the question as to whether it works. And basically, does the compound or new pharmaceutical entity do what it's supposed to do in the given indication? And these studies, they vary incredibly in their design because they're obviously based on what the indication is. If it's a cancer indication, if it's an infection indication, if it's neurological indication. At Legado Bio, I can go through the typical types of studies that we do in terms of efficacy models. And one such model is the xenograft studies, so they're cancer models. And in these studies, what we do is we have a mouse that we inject subcutaneously or orthotopically, that is to say in the organ in question. We inject them with cancer cells. These can be either murine cancer cells or human cancer cells. They're injected into the mouse. They're allowed to grow until they reach a specific size of tumor. Once the tumor has reached the size we want, the animals are distributed into different groups where each group has an equivalent average tumor size. And then they are dosed with the new compound according to what you have predicted the schedule to be from the PK studies. And over the course of the administration, we regularly weigh the animals to keep checking that this compound is tolerable, but also measure the tumor throughout the study. This is a very basic way to see if a compound works in vivo in a biological model, a biological cancer model. We also, oh, no, go back. Thank you. We also perform what we call vaccine or serology studies. And in a basic sense, they are, we dose a vaccine through the thought clinical pathway. So if it's an intramuscular vaccine, if it's an intranasal vaccine, subcutaneous, we dose this into the animals at the required schedule, usually once every 14 days. And then we assess whether an immune response is triggered either by taking serum for measurement of antibodies or taking spleen for measurement of cell-mediated immunity. These can also be combined with infections. So we work in a facility that allows BSL-2 and BSL-3 infections. So for example, influenza or corona. And we can do vaccine studies where we assess that a preliminary vaccine results in a inability for the animals to get infected. Okay. Thank you. So once we have seen that the compound works, it enters the bloodstream, we also want to know that the compound goes to where it's supposed to go and doesn't stay where it's not supposed to stay. So plasma PK alone isn't enough. We need to confirm that the compound reaches the target tissue. For example, if you have a compound that needs to go into the brain for whatever reason, for a glioblastoma or for an Alzheimer's indication, we need to confirm that it crosses the blood-brain barrier. We also want to make sure that it doesn't become clogged in target or non-target organs, such as the kidneys, which would lead to toxicity. This is where the biodistribution works and the ADME work becomes critical. So we want to look at both the distribution of the compound, but also the metabolism and the clearance to make sure it goes to where it needs to go and stays where it needs to stay. What we do in these studies is basically dose the animals as required, and using tools like LCMS, imaging, such as PET imaging or fluorescence or luminescence imaging, radio labeling, we can see either in a picture form or via measurement in the organs where the compound goes and how long it stays there. And this gives us a very clear foundation for the next and almost final studies that we need to perform. Next slide. So now that we know that the compound has good bioavailability, it's working in its indication, we're getting closer and closer to reaching going into the human, going into clinical trials. But before we go into clinical trials, we need to do something called a GLP toxicity study, which I will go into in the next slide. But before we do the GLP toxicity study, we need to do a little bit of risk mitigation in what are called MTD, or maximum tolerable dose, or DRF, dose range finding studies. So what we've done so far is we've looked at PK and we've looked at an effect, and we've basically looked at the effective dose. What we haven't looked at is the tolerated dose, so the dose limiting toxicity in the species that is relevant that we will be using in our GLP toxicity study. So to do this, to find a tolerated dose, we do these tolerability studies, basically, where we increase the dose to the maximum tolerated level. And by doing this, we find a dosing range that is suitable to go into a GLP toxicity study. That means the GLP toxicity study, which is a large, very expensive study, won't be designed inappropriately. If we design it with too high doses, then the animals may die from toxicity and the study is irrelevant. We can't make any conclusions. In the same way, if we use doses that are too low and we don't see any sort of toxicity, then we can't draw any conclusions from the GLP toxicity study. So we need to find a dosing range, and that is what these MTD DRF studies are for. MTD studies are generally single dose, where you give a single dose of test item and do clinical observations of the animals. Dose range finding studies are generally 7 or 14-day repeat dose studies, where you look at a limited number of clinical observations, including body weights, health status, and in some cases histopathology, to give you a good indication of what doses are tolerable and what doses we should use to go forward. Finally, when we're getting very close to our clinical trial now, we want to take our compound into phase 1 or phase 2, depending on the indication. And this means we are required to perform a regulatory toxicology study, basically to answer the question, is it safe for human trials? And these studies, they vary depending on the modality, depending on the indication, but they're all based on specific guidelines, ICH guidelines as they're called, or guidelines from the EMA, FDA, or WHO. Small molecules, they will follow the M3 guideline. Biotechnology-derived pharmaceuticals like monoclonal antibodies would follow the S6 guideline. Vaccines follow the WHO vaccine guideline. All these guidelines basically tell you how to perform a regulatory toxicity study to get your compound into clinical trials. The studies are basically designed so that they mirror the clinical setup. You use the same route of administration. So if you're going intravenous in humans, you go intravenous in your animal. You choose doses with a good safety margin so that it's a higher dose than what you think you were going to be giving to the human. And the number of administrations and the interval between administrations should also mirror what is happening in your clinical study. Generally, these studies require 2 different animal species. This can vary depending on what your modality is, what your indication is, but generally for small molecules, anyway, you would need a rodent and a non-rodent, and both of these species need to be relevant to your compound and the mechanism of action, basically. For things like vaccine studies, you only need one species. So we do a lot of vaccine studies because we're very good at working with rats, which we can use for vaccine studies. All studies generally need 2 sexes because most compounds are indicated for the entire population, males and females. So you need to do your study in 2 sexes. You have a main group where animals are given the compound according to your dosing strategy and are euthanized about 24 hours to 72 hours after the final dose. You also include a recovery group, which are euthanized 14 days or 28 days after the final dose to see if any toxicity recovers or if there is a toxicity that happens at a later stage. In these studies, you have your main endpoints, which is basically to look at tolerability, and doing that you do that by looking at body weights, health status, food consumption, so how much the animals eat. You look at what's known as clinical pathology, so basically hematological parameters associated with kidney function, liver function, metabolism, and urine analysis to look at kidney function and coagulation as well, to look at if there's any changes in the platelets or the coagulation patterns of your animals. You also look, in certain indications, you look at ophthalmology, so look at changes in the eye. And also, it's very important to look at histopathology, which is where at the end of the study, you remove all 40 to 50 organs from the animals, and these are all evaluated at the macroscopic and microscopic level to see if you have any compound-associated toxicity. These are the main endpoints. Depending on indication and modality, you can also include other endpoints that are non-safety related. So for example, toxicokinetics, to ensure that the compound has reached the bloodstream in your toxicity study and to be able to compare any toxicity you have seen with levels of compound in the bloodstream. When it comes to biologics, you can look at ADA or antibody, wow, my brain slipped there, ADA, I can't remember what it stands for. You can look at immunophenotyping as well in biologic studies. Serology or antibody production is an important parameter to look at in, for example, vaccine studies. And you can also add biodistribution, for example, with oligonucleotides or any gene therapy studies. You need to look at integration and distribution of your genetic material that you've dosed the animal with. So these are generally quite straightforward, standardized studies, but do have a little bit of variation depending on your modality and indication. And if everything goes well and you found that your drug is safe and tolerable in these studies, then usually you're able to go into phase 1 clinical trials. Okay. But it doesn't end there. There is after phase 1, there's phase 2, phase 3, and phase 4. And in these phases, you generally expand the population that you look at and look at both tolerability, safety, and effect in humans. And when you do these, you also need some other information that has to be provided via an in vivo study, particularly information about the effect on reproduction and the effect on a juvenile population if your compound is indicated for people under 18. What I have not included here as well is carcinogenicity. So some compounds you need to look at carcinogenicity by giving the compound. You can give it for over 2 years and see if the rats in this case develop cancer. These types of studies are advanced and are nothing that we perform at Legado Bio, but are important to later drug development. And I think that is all from me. So I hand it back to Matilda.
Well, thank you, Olivia. So having heard more about the scientific rationale for in vivo studies, I'd like to come back to who we are and where Legado Bio comes from. So Legado Bio is a young company, about a year, and located in the Stockholm area, but it's built on years of experience from lab animal research in Sweden. So originally fostered by the AstraZeneca, and more recently we have RISE in Vivo Lab in Södertälje, Legado Biomedical, and Scantox Solna in Vivo Services. All of these facilities have now been closed down. At Legado Bio, we have taken on the responsibility to carry that unique expertise forward, not just in methods and capabilities, but also in people. So several of the talented animal technicians who once worked with these sites are now part of the Legado Bio team. Lucky for us, lucky for you. So what we do is that we do not operate in a single facility. Instead, we work across several established facilities in the Stockholm area. That includes environments that belongs to Karolinska Institutet, Karolinska University Hospital, and also Stockholm University. So that gives us a flexibility, both scientifically and operationally, and depending what kind of project that the project requires. So it also makes it possible to match the study of the right environment rather than forcing everything into one setup. So when relevant, it allows also sponsors to work close to our study team. So that I think it's, we learn from each other, but also it improves the efficiency of the work. You do what you're best to in a project. So one of the most common questions that we get is which animal models and therapeutic modalities we work with. So there is no limit, we usually say, but we work mainly, of course, with rodents, especially with mice and rats, and because they are often the most relevant and efficient choice for early efficacies and mechanistic studies, also the translational readouts. So, but we also work with transgenic and immunocompromised models when it's needed. But some questions and some research also require other species, and it could be rabbits, ferrets, or mini pigs would be more appropriate for the study you have, and it depends on the anatomy, the physiology, pharmacology, the dosing route, the sample possibility, and the actual procedural setup. So the same applies with the therapeutic modalities. We can work with small molecules, vaccine, biologics, cell therapies, and oligonucleotides. Everything could be tested in vivo, and usually they are also needed to be tested in vivo, but not always the same way and not always the same species. So for us, we want to be part of the discussion from the beginning, and it's like always, the starting point is the scientific questions, and from that we take it further. So why are we relevant? I think we got the answer from Olivia's presentation part. But I think this slide shows the relevance what you working in the Bridge Sweden. So because many of the Swedish R&D companies and academic preclinical research groups do not just need a study, they need support of the whole part, and it's great when we can help out to find the practical and scientifically solution to move the project forward. So that's how we see our role. So in Sweden, where we do not really have large integrated CROs in this space, instead we have built it is a more collaborative platform, and where Legado Bio serves as the in vivo core. So we work closely, of course, with the breeders, but also with the animal facilities. But before we start the study, also where the test item are produced or synthesized, and the formulation experts, and what you get out from the in vivo studies, you need experts that do the bioanalysis, the kinetic evaluations, histology part, the histopathology. Some studies we could connect to people that are skilled in advanced imaging, and also for the studies that Olivia talked about, the toxicology consulting that could also be before our studies, but also be part of the evaluation of our studies. So so for all our early stage companies and research groups, I believe the strong network between service providers we have in Sweden gives both an opportunity and a kind of easy peasy solution. So moving forward quickly, but also gives you freedom to make your own choices. So also, not just as important what we do is also how we do it. And we work with a strong focus on quality, compliance, and careful selected partners. But we also, as I said in the beginning, place a great emphasis on animal welfare and refinement. So that is something that I would like to share with you now. I had the privilege of taking over videos developed by former colleagues from the Animal Lab in Södertälje, colleagues who did an outstanding job in developing and sharing refinement methodology. So I'm happy to carry that work forward and to continue showing a good practice can look like before and during an animal study. So of course, I know that every lab do not work under the same conditions, and not every study allows the kind of open environment that will be shown in these examples. But even then, there is usually something that you could learn from them, and something that could be adapted to your own research environment. So, because refinement is not only about ethics, it improves the robustness of and reusability and of your research data. So in more or less in our studies that we do, animals are ordered for the specific study. Often they come from qualified breeders in Europe, and that means that they arrive in a completely new environment. So in that way, they are unpacked and introduced to the new settings, and that would matter. So in this example, they are handled slowly and carefully without chasing, and giving time to settle in the hand. You usually you do your health check at arrival, and also to be sure that the sex is confirmed. And then you introduce them to a helpful tool called the vet bed, and that vet bed is kind of a snooty filth that comes through the whole study. It's a kind of where you could, something familiar, good to have when you are in a new environment. So in general, the animals are often around 10 weeks at the study starts, and it's good to have a long acclimatization period for the animals. For maize, they are usually good to have them longer acclimatized because it makes it easier to group hold them. So here is an example how you can work individually with a mouse and to prepare them for later study procedures. One of the most important things is that for us is never to lift them by the tail. It's not allowed, and you don't use a tweezer. And we encourage to use the hands. And as you can see, the vet bed is around that they recognize. And you saw the tunnel in the beginning. When they're new, it's going to be a helpful tool as well. So what you see here is a really great starting point for everything that you do later in a study. So in the next video, you will see oral gavage, oral dosing in habituated visitor rats. And these rats have been trained and gradually get used to the administration procedure that makes a major difference. They trust the handler so they can easily be picked up from the cage, and there is no need of firm grip, and you see it seeks the handler afterwards. It's a relaxful moment, and the animals get a reward afterwards. They are moved into the open boxes, observation boxes after dosing. So the soft tubes that you use is the same type of tubes that is also used for premature babies. And the same technique can be applied for mice. So this is, as I said, a super soft tube that can be used, but nothing comes without training. So if not trained, they will probably be chewing the soft tube. So you need to spend time with animals before you start. And maybe we also should have a look at the less successful scenario. Here we can see what the oral dosing looks like in an untrained or traditionally handled rat. It's difficult to restrain or pick up in the cage. It struggles. It vocalizes when held. And this rat does not trust the handle at all. You need a firm grip to dose the animal safely. And you can also see that the coat, the rats have urinated on the coat, and that's also a signal of stressful moment. Yeah, so you see the difference. It's obvious. So I think we will stop there. We kept the time. I hope that you now have seen a good view of what is usually needed when you move your test compound forward, and also have an understanding who Legado Bio is, and what I think is very important, why refinement matters. So whenever you're ready for the next step, we would be happy to continue this conversation. And yeah, we're ready to take questions, and thank you all for listening to us.
Thank you, Matilda and Olivia. We have a few minutes for questions. We got 2 questions in the chat already, so maybe we can start with them and then see if there are any questions from the audience. We had a question here that's, do you look for side effects during efficacy studies?
Yes, definitely. So, I mean, clinical observations of the animals is something that is done in whatever study we do. It's part, we need to know how the animals are doing at all times. So it is something that in times we don't officially record the daily health status of the animals, but if there is any adverse effects of the test item in any type of study that we perform, PK, efficacy, any type of study, we will record that. It's very important to keep a close eye on the animals. As Matilda has been saying about the 3 R's, it's very important to have happy animals and comfortable animals in a study. So we, of course, look at animal health in any study we perform.
Thank you. Another question is that in rodent studies, especially mouse models, it is well reported that the genetic background of mice affect the pharmacokinetics, difficult word for me, as well as phenotype severity. How do you select mouse strain and genetic background and its relevance to clinical setting?
Should I answer that, Matilda?
Yeah, do so.
Yeah. So that's a difficult question. When it comes to mouse strain, in terms of tolerability, toxicity studies, studies that you want to be clinically relevant, you tend to take, for general small molecules, you tend to take an animal model that is or a mouse model that is outbred. So there's different types of animal strain or mouse strains. There's inbred strains and there's outbred strains. Outbred strains have more of a genetic variability, and these are the ones that are used for tolerability because it provides a better assessment of tolerability. However, if it really depends on your compound and on your indication, if you have an efficacy model, for example, that only works in a specific strain, then you will be wanting to use that strain in your PK study, so it's relevant to your efficacy study. There are certain, I say we, in my previous job, we worked with a certain company whose compound was metabolized by a specific enzyme that was in mouse blood, but that did not exist in human blood. So in that case, we chose a mouse strain that had that specific enzyme knocked out to be able to provide a more clinically relevant PK answer, basically. So it's a difficult question to answer because it's very dependent on the situation, but it's something we think about in every study design, definitely.
Thank you. I hope that answered that question. Another question was, how do you translate the dose from rats, mice to humans?
I think it's still your part, Olivia. You're muted.
Yeah. So that depends. It depends on your what's it called? Your type of compound. Translating dose. That's not my expertise in terms of small molecules. I can't answer this very fully, but when we look at small molecules, for example, we are evaluating the dose in milligrams per kilogram. And there are specific formulas and equations to work out how your milligram per kilogram dose in a rodent or a mini pig or a dog translates to the clinical dose. I can't exactly answer that perfectly, but for certain compounds, it's milligram per kilogram. If for certain compounds, such as a vaccine, which is given as a whole dose, that will be basically the same dose given in animals as it is in humans, basically, the highest possible dose. So it's, I don't know exactly the answer to this question. I think it's something that I can look up and get back to you on, but there's people who are better at answering this question than I am.
Thank you.
Yeah, and I think that comes to the next question. Sorry for taking over here, but I see that we are short in time, and there is always a collaboration, and there is a number of consultancy services around us that we connect to both before the study and after the studies. And I see that we also have a question about the regulatory consideration. and designing studies. And we happy take part of these discussions and also could give guidance. Of course, as Olivia says, we have expertise in certain areas, and usually it's related to the expertise we have in research areas in Sweden. So we are good at cancer research in Sweden. We are good at vaccine. So I think it's kind of related what our expertise. Yeah, it's a network that we have, and what you have done before is we can help out with. Otherwise, we will learn and look it up, and otherwise we will connect you to the people that knows it best. But the question from Hamid about the guidance, yeah, definitely, that's where we can help out.
Thank you. I have one final question, if there's no other questions from the audience before we wrap this up. So I was wondering, so when in the development process do you think companies should start planning their preclinical in vivo studies, and when is the right time to actually reach out to a partner like Legado Bio to make this process as smooth as possible?
I would say as early as possible, because it's very, I won't say very common, but it has, I've seen it in the past where academics or other researchers do the wrong in vivo studies, basically, because they're not 100% sure of what is required in terms of in vivo studies to get into phase 1 clinical trials. And to be able to streamline the studies and to use as few animals as possible, because that is our goal, as well as to spend as little money as possible, because I think that is everybody's goal. It would be good to contact us as early as possible.
Yeah, I could just step in and say, I mean, you don't need to police the studies at Legado Bio. I mean, we could be a discussion partner also. And I think there is a great work that people and researchers do in their animal labs and research groups. I think the part where we come in that we do a difference, it's when you take the step from academic research and going into more industrial setting, and you need to have standardized way to design the study and standardized way to how to interpret the readouts, and you need to have the instrumentation around that also standardized. So that's where we will do the difference for in research.
That's great. Collaboration is key, and talk to you guys early. We like that.
Yeah.
So thank you so much for being with us today. And with that, I think we should end this webinar. And thank you everyone for taking part. We will send the inspelning,
Recording.
lost that word, the recording, thank you, to all the participants as well. So thank you and have a great day.
Thank you.
Bye.
Bye-bye.
Bye-bye.